for their support of the forum. We've heard really on and off in different settings about the importance of addressing new targets. And despite the amazing successes in coronary disease therapy in the last 2 decades, it still remains the dominant cause of morbidity and mortality in the developed world. So the next panel is really going to address new targets in coronary artery disease, and to lead that panel, we're fortunate to have Kathiresan, who's really been a pioneer in the modern genomics and genetics of coronary artery disease. He's currently the director of the Center for Genomic Medicine at Massachusetts General and also associate professor of medicine at Harvard. And he's really putting together the genomic causes of coronary disease with potential new therapies. So Sek, welcome to the podium and welcome to your panel.
Good morning. Thank you, Callum, for the kind introduction. We're here today to basically discuss the future of therapeutics development for atherosclerotic cardiovascular disease. As Callum just mentioned, we've made tremendous progress for this disease. Primary prevention, we have aspirin, statin, lifestyle, blood pressure lowering. For secondary prevention, you can add on antithrombotics, PCI. But atherosclerotic cardiovascular disease remains the leading cause of death worldwide. And we really want to discuss today how we can change that. So before we get started, let's have the panel members introduce themselves.
Aarif Khakoo. I'm the head of cardiovascular and metabolic discovery research at Amgen. I'm also the site head of our research site in San Francisco.
Good morning, I'm Clive. Meanwell, I'm the CEO of the medicines company.
I'm Paul Ridker. I'm a cardiologist at the Brigham and Women's.
I'm Campbell Rogers. I'm the Chief Medical Officer at HeartFlow.
I'm Craig Sponseller. I'm Chief Medical Officer at Kowa Pharmaceuticals America.
Okay, we'll start today with a question to be addressed by each member of the panel. So in an era of unprecedented LDL lowering, is there a role for additional medicines for atherosclerotic cardiovascular disease? Aarif?
Yeah. So my short answer would be absolutely. It's something that we think about a lot at Amgen, and one of the things that I guess I thought about in anticipating as you asked this question was, what do the numbers look like, right? So as many of you know, Amgen has just demonstrated data from the 4A trial that shows that PCSK9 inhibitors reduce, meaningful events like heart attack and stroke by, between 20 and 25%. And so if you take those numbers, just in the secondary prevention setting, and you imagine a world where PCSK9 inhibitors were used by every single eligible patient, you'd still have, in the US, about 1.5 million heart attacks and strokes annually. So should we as a community be satisfied with that? As a physician, as a cardiologist, I don't think so. It's not acceptable.
Clive?
Well, the answer is, of course, yes, but I think we have to throw a bit of a note of caution in there. We're not developing shades of paint. If you go to Home Depot and look for a certain shade of blue, they'll either have it or they'll mix it for you on the spot, and there's a diversity there which is extraordinary. And I think that's great for paint. It's not so great for drugs, though. And the idea that we're going to have a shade of blue for every patient is a bit out of reach right now, maybe down the road. So I think we have to be a bit thoughtful, because it's tempting from a financial point of view to go after those small target populations and assume you can charge a very high price. In other words, we develop an orphan disease program in cardiology and ignore the massive opportunities in population health. So while I do think we should be chasing genetic targets, and the people on the panel are way better qualified than I am to decide what targets those should be and what effector molecules we want to work with, I just worry a little bit that if we get too orphanish here, we're going to screw up the opportunity to change human health on a worldwide basis.
Thank you. Paul?
Well, Sek, I'm probably a little bit in between on this then. I look at this as the perspective of redefining what we call residual risk. For 30 years now, we've talked about residual risk as get the ApoB or the LDL lower. And clearly, as Fourier showed and as our SPIRE trial showed, PCSK9 inhibitors can do that. The effects were 15 to 20%. That's very important. And for some patients, that's a hugely critical thing. But the disease didn't disappear. So we've been talking about breaking residual risk into residual ApoB risk, residual inflammatory risk, residual risk on the basis of, say, elevated triglycerides, residual risk on the basis of, say, elevated Lp(a). And I suppose we could move into a genetic piece of that. The inflammatory piece is crucially important. Clive, you're talking about how many patients are at risk. There's actually more patients out there whose residual risk problem is a persistent pro-inflammatory response than a persistent elevation of LDL cholesterol, because statins are pretty good drugs. And so from a market perspective and from a biologic perspective, I think we're going to see a lot happen. We're seeing the innovation now, obviously, of novel ways of reducing Lp(a) for that group. And we'll hear a bit, I suspect, because we have Craig here from Kowa talking about trials that are being done with triglyceride-lowering drugs. These are big chunks. The paradigm shift is no longer everybody's going to get everything, but there's some pretty big chunks, and I think if we can target the right issues here, we'll find a middle ground that's going to work.
Campbell?
Yeah, I think the dilemma, and Clive really hit on this, is between the answer. So the answer is yes to your question's sake. The answer is yes. Clearly, there's a clinical burden which remains. The balance, though, as Clive said, is between identifying which patients really will benefit and therefore allow business models in the pharmaceutical world which enable and continued investment in research and development, and identifying and enunciating the value for these new agents and these new therapies, in which patients, over what period of time, with what endpoints. And clearly the trials are being done, but if you look at the current debates in the payer settings and in the clinical literature now around new agents, it really focuses on this. Is there value in which patients, and can that value be enunciated?
Craig?
Yeah, I heard yesterday some eloquent quotes, one of which was from Dr. Stockbridge saying that cardiovascular drugs is the graveyard of failed trials. That was an interesting one. And then Dr. Caleb today saying that we're suck and win when it comes to compared to oncology and immunology. Having said that, I think the question being, is there additional drugs outside of LDL-C lowering? It's certainly, we don't maybe need more drugs to lower LDL-C. I think we've heard yesterday that a third PCSK9 from Analem is going to be maybe a challenge, but certainly there's continued interest in lowering LDL-C. I look at it as, where should we as a company look at other areas, such as in triglycerides or triglyceride-rich lipoproteins, which we still don't have answers to. And through clinical trials like the outcome trial being done at Harvard with Paul's group, looking at that residual risk component, and maybe even more importantly, the inflammatory component is very, very much of interest. Others in other populations, like patients that maybe have other disease processes that don't follow the natural pathophysiology that we believe, like in patients with chronic HIV, they present a different picture. That becomes a very interesting question. And then certainly when we look at other populations younger, like in adolescent obesity, I think are also reasons why additional medications are going to be necessary, and we need to understand that.
Thank you. Paul, let's pick up on a comment you made. So in Fourier, with the addition of PCSK9 inhibition to statin, the LDL dropped to about 30. But at 2 years, there's still about 10% of patients who got both medicines who basically had an event, an ischemic event. Why is that happening, and how can we help those patients?
Sure. So at the recent American College of Cardiology meetings, I had the tremendous honor of going right after Mark Savage, and we presented both Fourier and SPIRE back to back. And I look at them as phenomenally consistent in what they're telling us, which is now we have a obviously proof of concept proven drug already class that clearly lowers LDL through a mechanism that's similar enough to what statins are doing in terms of upregulating receptors, that we have a safety signal that's very good, and it clearly works. And what SPIRE 2 said is if the on-treatment LDL is above 100, 20% reduction, and Fourier said for the entire group, 15 to 20% reduction. That's now established fact, and I hope it will change the ease of your and my prescribing these drugs so we get some reduced barriers for payers. That being said, we have to recognize who is the participant who's most likely to benefit. I've talked about the idea that what we really ought to be doing is selling the percent LDL reduction. Now, if I'm a payer, I'm very comfortable with that. So the drugs are designed to lower LDL. Show me it lowers LDL by 70%. I'll pay for the drug. If it only lowers it by 20%, I'm not going to. But equally important is what you're pointing out. There's residual real risk. And that, to me, says this idea you can just take LDL down to 0 and get rid of this disease, I'm not so sure. And it's very hard for me to imagine someone's LDL on a statin is 50 or 60, that the percent reduction going to drive it down is large enough. So that's why we're pursuing these inflammation reduction trials, one funded by the NHLBI using low-dose methotrexate, the other funded by Novartis using IL-1 beta inhibition. Those are pretty cutting-edge pieces of science writ large with 10,000 patients in these studies. And so that's a wonderful example of what I consider great academic industry collaboration to say we're going to take a risk and a gamble on a new biology. And that's why I'm sort of thinking we need to break out of this box and think about these other targets in a very direct way.
Great. And by the way, when are those 2 studies expected to report out?
Yeah, it's public knowledge that our Kantos trial, the canakinumab trial, is done. I am still blind, as is the sponsor. We'll get unblinded sometime late this summer, and we've made a commitment to try to present, if we can pull it off, at the ESC. It's an extraordinary science experiment. There's 10,065 patients worldwide on high-intensity statins. The LDL at baseline is 78. They're getting very aggressively treated. And what the conceit is, there'll be no change in ApoB, no change in LDL, massive reductions in IL-6 and CRP, and we'll see whether 25 years of biology is right or wrong, I guess, because the endpoint is fewer heart attacks, fewer strokes, or go home.
All right. So, Clive, let me pick up on a slightly different thread. Your company, the medicines company, along with Alnylam, is pursuing an agent, siRNA, against PCSK9, and are committed to both a LDL-based outcome trial as well as, sounds like, a large phase 3 that's cardiovascular outcome trial. But in general, I think Big Pharma is moving away from drugs for atherosclerotic cardiovascular disease. Why do you think that is?
Well, I'm not sure it's true. If you look at the massive investments that Amgen's been making, that Sanofi's been making, that Novartis is making in a different way, Merck is still making, it's easy to believe the short-term rhetoric that these big pharma companies are not any longer interested in investing significantly. Clearly, on any short-term cycle, a rational, emotional leadership team in a pharma company is going to say, what are our best returns in terms of impact on health and in terms of returns to shareholders? So that dilemma is there constantly. When trials don't go quite according to plan, there's a tendency for management teams to want to say, reassure investors particularly, that we're not going to waste any more money. And that's the short-term rhetoric that comes out usually pretty quickly after a setback. It's a reassuring tone for the financial community. It's a disappointing tone for the medical community. I think then it balances out in the long run, and you find leadership teams actually have greater complexity to manage than that. And I don't think you should believe everything you read in the press release right after a trial. I really don't. I'm probably the only oncologist in the room at this year's meeting, and by far the oldest oncologist in the room, if I am. And, I left oncology, moved to cardiology research at just the wrong time. My timing sucked, actually. Because I was chasing new anthracyclines and analogs of cisplatin in the 80s and early 90s, and we didn't have a clue what we were going after. The reason oncology became so fabulous is because we started to understand the biology, the fundamentals of the disease, and we're slowly unraveling that. And that led to forms of investment and clinical commitments that we'd never imagined previously. So, these are short-term cycles that you read about, but the long-term cycles are that cardiovascular disease is here to stay. It's the world's largest market. It's a huge opportunity for big pharma to make an impact. They need growth. So, I think the rumors of big pharma's exit from cardiovascular disease, much like you could say we did from antibacterials, not true at all. And I don't think we should discourage ourselves. We can sort of talk ourselves out of it if we're not careful. I don't think we should, and I don't think it'll happen.
Thank you. That brings up a good point about targets and new target selection. So one of the major challenges has been basically late-stage failures. A good example is inhibition of lipoprotein-associated phospholipase A2, daraplidib, 2 15,000-person trials, both failed. And these late-stage failures have been a real problem. So how are you thinking about therapeutic target selection and essentially de-risking targets and increasing the probability of success?
Right. So picking up, I guess, on Clive's thread, the challenge, the challenge in oncology 30 years ago was we simply didn't understand the disease, right? And obviously new science drove all of the new innovation that we're currently witnessing in the oncology space. Now, in cardiovascular disease and in particular atherosclerosis, the problem is that we just don't understand the biology, right? And if it's a very complex phenomenon. I can't model it in a dish, right? And the animal models that we have, simply are inadequate because they experience a form of the disease that, is super physiologic or doesn't even experience the primary thing that causes events, which is plaque rupture, right? So how do you understand atherosclerosis? And at Amgen, we have a deep conviction that it is human genetics that will lead us into the new mechanisms beyond non-HDL cholesterol to discover targets that could actually impact a very broad population with atherosclerosis. One example of this is we, in collaboration with our colleagues at Decode Genetics, just published a paper showing that the acyl-glycoprotein receptor, ASGR1, is that if you have a mutation in ASGR1, that you're protected from coronary disease, and the effect is not explained by a non-HDL cholesterol effect. So our belief is that success means starting with targets with human validation. Human genetics will give us that, and targets like ASGR1 meet this kind of profile, a non-HDL cholesterol effect, and evidence in humans that the target impacts the disease process.
That's fantastic. Campbell, as an interventional cardiologist, you were really there during the golden era of of devices for atherosclerotic cardiovascular disease, bare metal stents, and drug-coated stents. Where do you see device development going for this disease?
Sure. Yeah, it's interesting, and you're right. It was a golden era, but I think it still continues, although not so much in the coronary space. So it becomes and obviously the previous session with Mike Mahoney is about a really big, really successful company, although diversified, largely based on medical devices for diagnosis and treatment of cardiovascular disease. So it's clearly there. I think if one looks at where people are investing, both in big companies and small companies, it's really not much in the coronary therapeutic space, to be very specific in that particular application. If one looks at the sequelae of coronary heart disease in arrhythmia therapy, heart failure therapies with devices, all of those things are being deeply invested in, and certainly peripheral vascular manifestations. The cautionary tale for coronary investment is recently in the immediate present coming home to roost, is the current devices for treating symptomatic patients with coronary disease, which is a really large number, but is not the entire population of coronary heart disease, of course. That has been challenged with what's happened with the newest bioabsorbable stent technology from one of the leading, most innovative stent manufacturers, which with really significant investment has turned out to have relatively challenging clinical data in broad market use, and there's been a pullback from it. So that is probably even another bucket of cold water on anyone interested in investing, as you said, in the very specific notion of coronary therapies for symptomatic patients. But I would really think of it that way. The device world is wide open to entrepreneurial ideas outside of that particular application.
Great. Craig, I want to pick up on this biomarker issue. And LDL is a blood biomarker that is in the causal pathway for atherosclerosis, and therefore treating LDL leads to reliable reductions in disease risk. Do you think that there are other additional such causal biomarkers for atherosclerotic cardiovascular disease?
Yes, I would say for sure yes to that question. There are a lot of unanswered questions that we still have yet to elucidate the cause and effect. I think one of the big areas is related to triglycerides. Reason that we're embarking on this big cardiovascular outcome trial with Prominent using pemafibrate, which is a selective PPAR alpha, in patients with diabetes and elevated triglycerides and low HDL-C, particularly below 40 milligrams per deciliter. The important part is to understand, and knowing we've had previously failed trials, what is the value of treating and lowering triglycerides or remnant lipoproteins in this population to curb cardiovascular events and morbidity and mortality. The evidence is there that when we look at randomization or Mendelian randomization studies, particularly the large one out of Denmark, 70,000 subjects, showed that if you do increase VLDL cholesterol by 39 milligrams per deciliter, that significantly increases the cardiovascular risk. Therefore, we have to embark as a company to try to engage in answering questions like and persistent with, should we treat triglycerides and is it the right focus? So I think those markers or those biomarkers are going to be very important related to remnant lipoproteins and triglyceride-rich proteins. The other is looking at macrophage activation. I think it's really critical. When we heard yesterday about connecting immunology with oncology, and that was sort of the renaissance and the breakthrough, there is also this application of understanding macrophage activation and trying to intercede and prevent those pathways to lead to that. And we have a very nice relationship and a collaboration with the Center for Interdisciplinary Cardiovascular Disease here at the Brigham and Harvard with Kowa to understand and pursue those types of things and understand macrophage activation, plus other things related to calcification and insulin resistance, which I believe are also very, very important markers. And lastly, I think, as we've embarked on looking at chronic HIV, I think there's some important questions to answer here based on the pathophysiology that we have. It's important to understand, are there other markers of inflammation and immune activation that we should be looking at that will help better treat disease that's not necessarily identical to what is in controls or non-HIV patients? Looking at things like soluble CD14, which is a subclinical atherosclerotic marker we should be looking at. LPPLA2 and is a non-calcium dependent lipase that's creating active changes on the surface of the LDL that are very oxidized, very high pro-inflammatory, looking at that as well as oxidized LDL, which is the most pro-inflammatory. Given all of that, I think that these are areas where we believe we need to continue to look at because they are at risk, and these are markers that we haven't actually found targets for.
Thank you. Let's do a question for everybody. I think one of the big elephants in the room is cost of drug development. How long it basically takes, but also the amount of resources to get a target and a therapeutic screen, a molecule, all the way to clinical practice. How can we decrease the costs? Arif?
So this has been a big topic, I think, at this meeting, and I think it's a really important topic. And there's lots of good ideas. You know, figuring out ways, adaptive trial design, for instance, is a reasonable thing that could be applied very well. That we could compress trial time, increasing our understanding of risk, right? So that you can enrich trials with higher event rate populations. That's something that can certainly, again, reduce trial size, but. You know, I just want to, if you look at the broad picture, right, and there's actually a very nice parametric analysis done of this. So what drives trial efficiency, right? So the amount of money you spend over time, right? And there was one of our peer pharmaceutical companies actually published this very nice paper looking at, sensitivity analysis of if you change certain parameters, cycle time, trial size, cost of trial, where do you get the most bang for your buck in terms of operational efficiency? And it's all around the probability of technical and regulatory success. That is, fail less. So our belief simply is that you have to pick the right targets. That will reduce costs because there's nothing more expensive than a failed drug trial. So, and that again is why we have this deep conviction in starting with targets with human genetic validation.
Thank you. Clive?
I think that's very important. I'd sort of say that many people are working hard to try to reduce the costs of the operations of clinical trials and pick the right targets and so on, and use statistical methods that allow sampling instead of 100% source document verification. You can go into the minutiae of the process of running a clinical trial and I think find some meaningful savings there. You could also ask the question, is it really a question of cost or is it a question of productivity and efficiency? And one way to make clinical trials more cost-effective would be to include new members in the coalition of designing clinical trials. I think that we can take a lesson from other industries such as Boeing, who involve Lufthansa engineers in the design of their airplanes. When we come out of the FDA waiving our approval at payers, we tend to say, instantly, I got 500 planes built at the back, do you want to buy one? And the answer is not always yes. It's sometimes, it's not efficient enough, I don't want to buy them. So I think we need to involve decision makers who make economic decisions in the design of our phase 3 programs. And it's been hard to do because the skill sets are not evenly distributed. And the willingness to do it hasn't necessarily been there. But it now is, I think. And I think now's the time to ask, whether we can build more meaningful economic endpoints into our phase 3 programs, not waiting for approval and then building them into some kind of quasi phase 3B, phase 4 program. So I would like to see the efficiency and therefore the cost of trials come down because we actually ask different questions and include additional stakeholders earlier.
So this partnership with payers kind of from the study design perspective, are you trying this for the
Yes.
siRNA?
Yeah, absolutely, because we are very fortunate to be able to learn from the work of Amgen, the work of Sanofi, the work of those who did statin programs. What we're very interested in also learning from is the pressures that those other stakeholders are trying to manage and whether or not they can come to the table. They may not have expertise in designing clinical trials. That's not the question. But they may have real helpful input into what we think about designing clinical trials. And I think without those people at the table, you're going to come to this moment of crunch after you've done the work where you have to decide whether the work was meaningful to them. And it may or may not be.
Paul?
So, Sekar, I'm in the camp that probably would like to blow up the entire clinical trial system, despite the fact that I run a lot of these large-scale trials, because I think if you run these, you see the inefficiency across the board. So first I'd say, I think we need to readjust so that every patient in every hospital in the United States is part of a clinical trial. I mean, it's an admission that we really don't know what we're doing, and we have better ways to get there. And I think patients would buy into that very simply. I said tongue-in-cheek at a recent NIH board meeting that if we simply told the deans of every medical school in the country that you're not going to get your R01 money if you don't put people in the clinical trials, everybody would redo everything instantly, and we would enroll all of our trials in North America in about 3 weeks. So there's a way to get there if we thought about it. Now, what we won't abandon is randomization. That is the absolute crux of the issue. And I would probably argue we're not going to abandon double blinding either. Those two things give us validity and accuracy. So the question is, how do we change this structure to guarantee the two most important things, but get rid of the things that are just frankly in the way? Now, Aarif was talking about using approaches that accelerate the net risk, increase the absolute risk. I would argue, yes, you want to do that, but make sure you amplify the risk on the basis of the biology of the intervention you're interested in. So all of our studies are biomarker-driven enrollment criteria where the say, It's an inflammatory biomarker because the drug is going to reduce inflammation. Inspire 2, it was high LDL because that's what you're trying to drop. When I sit in boardrooms and pharma designs a trial, say, well, we want to use like a Framingham score or increase risk, I just roll my eyes and go, oh, no one's learned anything. You've got to enrich the population, but to de-risk your program, make sure you know the biology and then follow the biology. Don't get diverted into other areas. So I actually am optimistic. I think costs could dramatically come down these trials, but it's going to take a desire and a willingness to do so and some creativity at a regulatory side.
Campbell?
Yeah, I think that the ideas up here are from incredibly experienced people with clinical trial design, and they're really thoughtful about ways to shorten, abbreviate, make more efficient clinical trials. I would take perhaps a slightly different orthogonal perspective, is that the goal of these development programs is, of course, to find better therapies, but it's also to find things which can then, for the sponsors, be monetized. And I think the approach which perhaps has been lacking is to think holistically about a population and how that population will be managed, and take that sort of intellectual approach right from the very beginning. And yes, it's about finding patients at risk. That increases event rates, makes these sample sizes smaller and so forth, absolutely. But also thinking about that same population from the net spend on cardiovascular disease, which they are accruing, which they are creating as patients. And just as an example, and thinking about it holistically, right, so that the drug and the patient and their journey are considered as one, even in the trial design, so that then, at the end of that trial, one can say, here's the clinical value, but also here's the economic impact in a way that's not just, we're adding this drug, we're lowering LDL by a certain amount, and over time, these will be the perhaps savings related to that. These are, in current trial design, sophisticated health economics methodologies, these are incredibly measurable things. And that's an element which, were it brought in earlier, it wouldn't necessarily reduce the costs of trials. So slightly different from your question's sake, but it would allow the outcome of the trial to perhaps speak more to the current world we're moving to, which I know has come up here, of creating value and enunciating value for a new product.
Craig?
Yeah, I think I'm faced with some challenges because I'm not as large of a company as my colleague over here to have a very exorbitant pipeline that might be of relevance as we pursue cardiovascular disease. Having said that, I think we have to be due diligent as a moderate-sized company to say what is the right place to put the dollars and the resources. I think it's important questions that have to be answered. That's why we've spent a lot of time saying, is it still important to address the question about triglycerides in patient populations that have diabetes and high trigs? We believe that. It's taken a long time, but I think Paul's right that it takes a collaboration and a communication as to how do we make the costs very effective to answer the question based on the biology in the most relevant way that it matters in the end that we've done something very different, and certainly proven a hypothesis I think we all believe currently still. The other is to try to help differentiate. If we have a product, Kowa has a statin, pitavastatin, if we're trying to stay in front of it, we have to be able to help differentiate. And a lot of times that's a topic hard to address, because how do you differentiate statins? And there are some areas, and one of which is that we're looking at in terms of insulin resistance, and in certain populations. An at-risk population would be HIV. So with the money invested in a Reprieve trial, which is an outcome trial for the first time, because there's many questions that we haven't answered yet about that in cardiovascular disease, is there certainly in an at-risk population for incident diabetes or glucose intolerance that we don't have an effect as a statin versus what might be happening in other statins, which we've seen some evidence of. In the Saturn HIV trial, a little bit different in terms of insulin resistance and insulin sensitivity. So we have to be mindful of that. And I think in the future, are there other areas to look at real-world data that might bring costs down to still answer questions about what we don't know? So I think that's something we still have to think about as well.
We'll pick up on a related topic about surrogate endpoints. So we talked about LDL, we talked about potentially triglycerides. There's a lot of advances recently in terms of non-invasive imaging of coronary atherosclerosis using CT. And maybe Campbell, is there a role for coronary plaque as determined by CCTA as a surrogate endpoint in terms of in the drug development process?
Yeah, that's a really interesting, and it's a provocative question. And there are certainly people in the room here who have a lot of experience in that from partners in terms of using CT and imaging. As heart flow, that's what we do, looking more at symptomatic patients, so not quite to your question, but using CT and saying, can we derive, what information can be derived from that that's helpful? I think the current world of cardiac CT with sophisticated imaging and good technique is allowing imagery not only of coronary anatomy, but of plaque composition, which is really sort of mind-blowing, even from a former interventionalist, as you said, where the invasive angiogram is everything. Coronary CT is pretty much there, and more so because, of course, one can see the plaque. As a surrogate endpoint in trials, it's already in place in a variety of trials, and we're actually involved in some looking at both CT and also flow implications of stenoses and different progression and regression. Of course, as Paul said, the rigor of randomized trial, double-blinded, and hard clinical endpoints in the cardiovascular therapies world, those are so important, such important three legs of the triad. It's intriguing to think about surrogate endpoints like that for really big definitive trials. Certainly in early stage trials, yes. I think it's going to be a little bit longer slide to say we're going to use that as in lieu of outcomes in any kind of real definitive way. The final thing I would say about imaging, of course, is the little known fact, or the little talked about fact, is that there are massive amounts, back to the value-based drum, which I keep hammering, is there's massive amounts of money spent, hundreds of millions of dollars on imaging today, much of which could be done more efficiently. So again, back to the trial design, thinking of how imaging, novel approaches, more efficient imaging of coronary disease is a way to free up in the system hundreds and hundreds of millions of dollars, which today is being spent on patients, many of whom don't actually have coronary disease because they're being done because there isn't a better test. So back to the thinking of how can we free up funding in the cardiovascular world for innovation, that is another potential avenue that you mentioned.
I think another general theme is kind of stratifying patients and identifying subsets that will basically still be at risk after standard of care. And so how are you thinking about that in, for example, the ASGR1 program? Right. How are you going to select out the patients you think most likely they're going to benefit from this new mechanism? Paul's talked a bit about using blood biomarkers, but are there other approaches to think about stratifying patients?
Yeah. So I spoke about the opportunities for using genetics as a starting point. Now I'll tell you a little bit about the challenges, right? And that is that we have you start with this completely unbiased view, right? So you don't really understand the biology. And it could be that things like ASGR1 are completely touching on these inflammatory pathways that Paul's mentioned that are there's clear signals, right? So the starting point for us for any of these genetically derived targets really is to start from that and then go back and understand the biology we recognize at Amgen that it's a community effort, right? So we are as excited to hear about academic scientists in the field that are working on this specific target that resulted from the genetic finding to really understand the biology of ASGR1, because the biology really will drive understanding of the patient population that would benefit from the therapy. Although the genetics suggest that it has a protective effect in an unselected population, which would suggest that the effect would accrue across broad populations, but certainly understanding this on a biology perspective I think is very important. I think the second thing is, again, things that can just increase event rates. So how do we treat sicker patients? And whether it's a combination of phenotypic analysis, better risk scores, or genotypic analysis, a genetic risk score, which we spoke about, or some combination of that, a phenotype-genotype combination, just to simply focus our therapies on patients who are more likely to get events. I think that is an enormous challenge and probably one of the most important questions in the field, in my opinion.
Anybody else on the panel want to add to that?
Yeah, I think one of the more understudied biomarkers is the number of dollars that patient's going to cost you for the next 10 years. And we kind of ignore that at our peril. I think that there are data sets now that allow you to tease this apart and say, where is my money going to go if I'm caring for these patients or providing for these patients over the next 10 years? And it's interesting to me working with some of the most advanced healthcare systems in this country and around the world, places like Manchester, places like Intermountain Health, where you would imagine that these things are well known. You would believe, and yet there's very fruitful research happening there to try to identify where does the money really go.
Yeah.
As a biomarker, expensive patient, cheap patient might sound rather cruel, but it might be an important thing to consider before you start the trial and decide who you want to enroll in the trial. So now I'm going to come out as the bad guy, but that to me is something we can't ignore.
So, Sek, I'm going to come back to the genetic question because I want to ask you the question. You're more expert than anyone on this panel, really, in fundamentally the genetics of this disease we call atherosclerosis. My bias here is that the genetics is a genius way of getting at extreme phenotypes. We find these unique patients with unique issues with extraordinarily either advanced disease or, on the other end, no disease, and then we learn that biology, and then we try to apply it broadly to the population. But you and I have both been in this genetic risk score business for a long time, and while the p-values are exceptionally significant, the magnitude of the risk is moderate. And so I'm interested in how you see that in terms of taking these things to trials, or are we going to stay with the biologic model predominantly?
Yeah, I think the biggest value of the human genetics is to really expose new pathways that we previously did not understand for risk for coronary artery disease. And I think the genetics has kind of led us there. So the unbiased studies have revealed what we already knew. LDL is important, ApoB-containing lipoproteins are important, blood pressure is important for coronary artery disease susceptibility, but also has exposed a range of additional pathways that I think over time we'll understand better. So I do agree with you, the biology is most important. But it has been interesting over the last couple of years where the susceptibility, when we think about why somebody gets risk for heart attack, particularly early in life, it can either be due to a single mutation that kind of pushes them. But for most patients, that's not the case. It's actually the additive effect of maybe hundreds of thousands of DNA variants, each with a small effect. And when you sum up all those variation into a score, you can get pretty significant gradation across, and now we're seeing maybe 5 to 6-fold difference between the bottom quintile of a polygenic score and the top quintile. And that polygenic score is completely independent of LDL, blood pressure, and so forth. So I think, and what we do with that information, time will tell, but recently we showed, for example, that those at high polygenic risk had the greatest relative benefit from statin therapy, which was very surprising. We did not expect that going in. That was in Jupiter and ASCOT and also WASCOPS. And so this now is the first subgroup ever identified for statins that seem to have greater relative benefit compared to any other subgroup. And so again, how we implement that in practice, and is that useful information to select patients who might benefit from early statin initiation, that's all time will tell. But in general, I agree with you that basically new biology is the main value of genetics.
Right, so I asked partly, Sek, because I'm fascinated by your own recent data that when we isolate people with LDLs above 190, the number that we tell our clinical colleagues is likely to have FH on a genetic basis. My recollection of your elegant data was it's 2 to 3% actually had a genetic defect. The vast majority just have these multiple reasons for having high LDLs. And so in the clinical realm, it boils down to what's your LDL. But I agree with you. The polygenic approach is therefore more promising. But I think it's fascinating that the place where we have the best genetic data, which led to the PCSK9 revolution, is actually a trivial fraction of who actually has a high LDL. So I thought it was really instructive paper.
Yeah, thank you. Well, let's close in the last few minutes with kind of a rapid fire. I guess I'd like to go to each panel member and basically ask what you think is the biggest challenge to therapeutics development in the cardiovascular space. And then we'll come after that with your suggested solution to that challenge. Arif?
we talked a little bit about sort of targets sort of in the very early part of the process, but I do want to sort of switch gears a little bit to something that we think about deeply, and that is this other part of a very important conversation that we're all having about value, right? And the importance of the value of medicine in terms of combating what's a big societal problem, that is rising healthcare costs. And one of the things that's pretty clear is that what drives rising healthcare costs more than anything is disease, right? That is the enemy. It is the reason that healthcare costs rise is because heart attacks continue to happen, cancer continues to happen, and we have to care for Alzheimer's disease continues to happen, and we have to care for these patients. So we really fundamentally believe that medicines are actually an important part of that solution, not part of the problem. And I think the challenge that we're all facing, and I think is going to be part of an evolving conversation, is we believe deeply in this notion of value-based pricing. And the question is how to quantitatively understand value in a way that we can all agree is a good return on society's investment. I remember I was a medical student when statins first came out, and there was this deep discussion. Boy, statins are so expensive. That was the discussion. And you look back on it now, and I would argue that it was one of the best investments that we made as a society for human health. And so I just think it's very, very important in a world, especially in this current political context, we're very focused on sort of short-term things and not looking at the long run, the environment, for example, that we really need to sort of keep in mind the long view of this, and that is that the enemy is disease.
Clive, the biggest challenge you see?
Well, look, we're all afraid for our intellect, for our future. History would suggest that we've overcome those fears by moving from self-interest to slightly enlightened self-interest. There's not a big difference between those two things in personal behavior, but there's a whole lot of difference in economic outcomes. So I think we've got to move from self-interest to enlightened self-interest. I think that the stakeholders have got to get to the table. Mexican standoffs have to end between payers and vendors and entrepreneurs. And until that does, we're not going to make much progress. But I completely agree that bringing the right people into the room is what we need to do.
Paul, the biggest challenge?
For the sake, you're going to have to forgive me for being a little bit problematic here, but I grew up in Washington, D.C. in a family that had political connections, and my parents were in government. I think we better double down on science. And I'm very nervous about that. I know we're going to have a nice discussion between Gary Gibbons and Betsy Nabel coming up. I think that even though I'm a population biologist and epidemiologist who thinks about vascular biology, I think if I had limited resources, I'd keep it in the basic science realm. I think you got to get the fundamental breakthroughs, and then the rest of the stuff happens. But if you don't do the basics, and if we as a society don't know what facts are and can't understand science, we're in big trouble.
Now you have to say something that generates applause.
Right, exactly. That is a high bar. I'm sure this will not. No, I think that, and we've talked about it, and these guys have talked about it, that having the stakeholders, including the payers, in the conversation very early will overcome these challenges. And in particular, I'll give you just a factoid to just exemplify this. If you look at the patients who today go for a cardiac cath, for chest pain, about 2/3 of them don't end up having a PCI or a bypass operation. That's a fact. It's a fact here. It's a fact around the world. It's not because people are doing it willfully. It's because there's no better way to identify. And then think of the money being spent on those caths. It's hundreds of millions. Now, globally, it's billions of dollars. Now, if you say, okay, that's money being, in effect, wasted, which ought to be perhaps spent as part of the rising healthcare costs, and that's not being driven by disease. That's being driven by practices which are perhaps outdated or need different technologies in order to replace them more efficiently. So thinking of it holistically will overcome those challenges, but without that, it's hard to see how it doesn't kind of hit a ceiling.
Craig, you get the last word.
Yeah. Is this problem and solution or just problem?
Both.
So I think one of the main things I think is absolutely critical in the paradigm of pragmatic clinical trial development, I think the inflection point has to be moved far up from the point of basic science research to that of clinical relevance and clinical application. I think that that has a lot to do with identifying meaningful novel targets, first-in-class opportunities, but it has to be able to be in line with that you have the development or you've got the target discovery and validation, but at the same time you're beginning the assays and you're beginning the preclinical and you're moving faster to clinical development. I think that is really important for us. That's cost effective. We've partnered very nicely. I think it's a very important partnership between academia and the industry to do such a thing. Professor Kowa has been very instrumental in creating such a model so that we can advance faster. And I think it's meaningful to all these targets that we've been talking about to get them quicker to the patient and into practice.
Well, I want to thank the panel with a nice round of applause. And have a great day. Thank you.